Okay, so yeah, thanks so much for having me. I'm glad to be here today. We had to, or I had to cancel this talk last year because I completely lost my voice, so I feel bad for having to cancel. And I'm also happy to be able to do it on a Thursday because I teach Wednesdays. So thank you for everyone's flexibility on that. Um, as Anna mentioned, I'm going to talk about our Lake Champlain Basin grant, which was looking at the trends and drivers of mercury bioaccumulation in Lake Champlain. And I'm going to change slides somehow. Hold on. Oh, all right. I'm going to get this. Okay. So what we have here is a cycle of how mercury moves through the environment. I wanted to get everyone riled up by starting with the biogeochemical cycle. So mercury is fascinating. I've been studying it for a long time. It's the only metal that has a liquid, a dissolved in a particulate phase, and a gaseous phase. And so it has a really complicated global cycle. It's important to note that mercury is a global pollutant. We often think of contamination as something that comes out of a smokestack or something that comes out of a pipe. Um, in fact, for mercury, often pristine systems have the highest mercury concentrations due to long-range transport of mercury and ecosystem sensitivity. So, to quickly run through this cycle, mercury is released into the atmosphere largely from human activities, particularly coal burning and metal smelting. And it travels long distances in the atmosphere in its gaseous phase. It's then deposited on watersheds by precipitation and uptake by vegetation. And when it gets into aqueous environments, it can be transformed to methylmercury. And methylmercury is the toxic and bioavailable form of mercury. And so that's actually the mercury that we're concerned about. Some of that mercury is then taken up at the base of the food web. So phytoplankton at the base of the food web absorb methylmercury, and then mercury biomagnifies up the food web. So zooplankton eat phytoplankton, small fish eat zooplankton, big fish eat small fish. And at each of those trophic layers, mercury concentrations get higher. Fish take that mercury in, but they can't excrete it very efficiently. Um, in looking at this very small food chain at the bottom of this slide, you'll note that one of the biggest jumps in mercury concentration is between water and phytoplankton, and then phytoplankton and zooplankton. Those are very important to mercury bioaccumulation. What happens at the very bottom of the food web really predicts the concentrations in those big fish that we eat at the top of the food web. So in terms of reducing our exposure to mercury, there's really two realms where we can control our mercury intake. Um, the first is emissions reduction. And this is really important. The Clean Air Act had a big effect in reducing the emissions of mercury into the atmosphere in the United States. The Minamatic Convention is the global treaty, which a number of countries have signed to reduce mercury emissions globally because they affect mercury travels around the world, and so the global convention is really important. And then the Mercury and Air Toxics Standard is specifically to remove mercury from coal burning plants in the United States. On the other end of the mercury cycle, the other way that we can protect ourselves from mercury exposure is through public health. And so that means particularly providing fish consumption guidelines. In between those two things, though, there are a lot of ways that perturbations of an ecosystem can affect the cycle, or can affect the mercury levels in fish. And so, changes in how much watershed input of mercury, changes in the food web can affect how much mercury ends up in fish. So Lake Champlain is a fascinating system in which to study mercury, and for a few reasons. As we all know, it's a series of interconnected basins which have different properties. They have different depths and they have different connectivities to the watershed and different trophic statuses. So there are clear deep basins, which are similar to the Great Lakes or to the ocean, and those have very efficient mercury bioaccumulation from the water, so from the water column to the phytoplankton. By contrast, some of the shallow eutrophic basins, where there's really high algal concentrations, they're getting a lot of mercury from the watershed. Um, and the high algal concentrations can actually dilute the concentration of mercury at the base of the food web. So if you have a lot of algae, then the concentration in the algae itself can decrease because they're sucking it all up from the water, and that limits their uptake. Lake Champlain also has a really large watershed compared to other water bodies. And so that means that it has a lot of watershed influence. So it makes it a really interesting place because we have all of these different basins that behave a little bit differently, but within the same lake where we can understand how mercury gets into fish a little bit better. So the Lake Champlain Basin Program over the last 30-ish years has conducted game fish surveys of mercury about every 5 years. And these have been conducted by different groups and by different researchers. It's quite an extensive study. They look at 5 different species, and each of those species, they're looking for 10 individuals per basin, so from each of the 7 basins. And so I conducted the 2022-23 fish collection. Um, and I got white perch. So there's 5 species: white perch, yellow perch, smallmouth bass, walleye, and lake trout. And we got 10, we aimed for, we were a little bit short on walleye, but we got 10 individuals of each of those species from each basin. The lake trout, of course, only live in the deep main lake basins. And I want to just start by giving you a quick look at the data altogether. Jumping to the data. So these are the concentrations of mercury in all fish from the 2022-23 study. These data are as is, they're uncorrected for length. Um, so note that the larger predatory species, these are larger fish, they have their higher trophic levels, have higher mercury concentrations, those being the lake trout, the smallmouth bass, the walleye, they're higher on the food chain, they have higher mercury concentrations than the white perch and the yellow perch. And so those are the species that we maybe want to limit our consumption of, particularly, or the frequency of our consumption of, particularly for the really large individuals of those species. Whereas the white perch and the yellow perch have much lower concentrations, and so those are better choices for eating consistently, for eating frequently, multiple times per month. The other thing that we should note here, though, is that size has a really big impact on mercury concentrations. So within species, these are lake trout. You can see that we caught fish over quite a large size range. And that as size increases, mercury concentration also increases. So the larger fish take more mercury in and they excrete it very slowly. So they're bioaccumulating more mercury into their tissues. So for consumption choices within the same species, smaller fish tend to have less of a load of mercury than larger fish. For purposes of analyzing the data, this relationship with length can be kind of problematic, because there's a covariance between length and mercury. So if we wanted to look at like what are the differences across species, then we have to somehow take into account that factor of length. And it's problematic because across years we sometimes had different length distributions that we then have to account for. Um, but also because we're a little bit biased by species. So when I did the collection, we got a lot of our bass and our smallmouth bass and our walleye from fishing derbies. And I did this non-lethally. So I took a biopsy from a live fish and then analyzed that for mercury. People at fishing derbies are really only bringing in their prize catches. And so we were really biased towards large fish in those species, but not so much in the lake trout and the perch, which I got through the Vermont DEC surveys. So the size does cause a little bit of a skew in this data. All right, so what did we find? These are the temporal trends of mercury across years in each of the 5 species. So across the top, we have the yellow perch, smallmouth bass, and lake trout. surveys, like the survey years vary a little bit. We had to lump those early surveys because there weren't very many fish in some of them. And so we have fish from the late 1980s, sort of mid 1990s, and then going up to 2023. And generally the last three surveys, sort of 2011, the 2016, and then 2022, had much better numbers of surveys of fish than in the earlier surveys. But what we see is a fairly interesting trend. So across the top row, we have yellow perch, smallmouth bass, and lake trout. And those are fish that inhabit really different parts of the lake. So yellow perch are small. They're feeding mostly from pelagically or mostly from the water column. Smallmouth bass they're much larger. They're warm water fish. They're feeding littorally, so from around the edges of the lake. And then lake trout are these cold water, deep fish that only inhabit the deep basins of the lake. And yet we see this kind of interesting trend across all three of those very different species, which is that starting in the late 1980s and up until 2011, there was a decline in mercury concentrations in fish, which is great. And that's kind of what we would hope for with improving mercury emissions around those years. And then in 2016-17, we see this bump where concentrations increase, in some cases quite a bit, quite dramatically. These are mean concentrations across in species. I should have said that at the start. These are mean concentrations that are length corrected. So the mean concentration of all fish in the lake increased between 2011 and 2016 for these three species. And then in 2022, we came down to about 2011 levels of mercury again. The other two species that we surveyed, the white perch and the walleye, didn't really have those trends. White perch are a little bit bigger and a little bit more benthic feeding, where they feed like more from the sediment than their yellow perch counterparts. Not much of a trend in location or over year for the white perch, although like 2022 was the first time that we saw an appreciable decline in mercury concentrations in that species, which is great. And then walleye are just large migratory fish. There's not much of a trend there. We had a really big distribution of lengths in those fish, which make them a little bit difficult to analyze. And then one more trend just to look, so this is a little bit of a confusing graph. These are each of the basins that we looked at in the seven basins of Lake Champlain. And then in each one, there's three dots, those being the mean concentrations from 2011, 2016-17, and 2022-23. And a couple of things stood out. One is that Mallett's Bay in all years, that's that really oligotrophic basin, had higher mercury concentrations than in yellow perch than the other basins of the lake. So yellow perch are probably the fish that have the most localized response to mercury. They're the least migratory. They're feeding from where they are, and so we tend to see more of the basin effects in yellow perch than those other fish. And so that replicates what we've seen in previous studies of mercury and zooplankton, that Mallett's Bay has much higher or more efficient bioaccumulation, compared particularly to Missisquoi and Northeast Arm, which are those really eutrophic basins. And so there, the mercury bioaccumulation is quenched by those big algal blooms that can occur in those basins. And then the other thing to note, or that we noted across these 3 years, and this was pointed out in a paper by the researchers who did the previous survey in 2016-17, was that little spike that we saw across the three fish species in 2016, so that jump in mercury concentration actually only occurred in the deep basins. So it was only a significant change in the deep basins, and this was true for yellow perch, smallmouth bass, and the lake trout, which only inhabit the deep basins. So what is going on here, and why is this? Um, Over the past 20-ish years, The Mercury Deposition Network, run by NADP, the National Atmospheric Deposition Program, has monitored mercury in rain or in precipitation in Underhill, Vermont. And they've seen very little change. There's been very little change in mercury deposition at that site over the past 20 years. To get at longer records, one of the things that we can do is look at lake sediment, or in this case, peat bog archives. And so peat bogs and lake sediments can record the amount of mercury that's been atmospherically deposited. Um, and so in 2024, we took a core from the peat bog in Maquam in the Missisquoi Refuge, and then reconstructed. We used geochronology to date each layer of that peat, and we reconstructed mercury deposition. And so we can see how mercury was being deposited to the lake watershed over longer time periods. And what we note is that following the Industrial Revolution, there was a steep incline in mercury deposition in the region. And then following the legislation of the Clean Air Act, mercury and air toxic standards, we see a reduction in mercury concentrations in what's in the atmosphere and what's being deposited. And then a little, I don't have error bars on here and I should, but it sort of flattens out in the more recent years, or it's a little bit hard to tell. And so we should be seeing that in some ways replicates what we're seeing in the fish, that there was a big decline in mercury concentrations, although we kind of expect it to continue to decline in recent years. A similar thing has been noted in other lakes in the Northeast, and particularly in the Great Lakes. And so we often compare Lake Champlain to the Great Lakes. It's in the same ecoregion as the Great Lakes. And so they sort of make a nice comparison. In the Great Lakes, we see that these are just the lower Great Lakes, so Ontario, Michigan, and Erie, and these are concentrations of mercury in lake trout surveys from those lakes. Um, Between 1980 and 1990, there was a decline in mercury concentrations, and then it seems to have really flattened out, that we haven't seen further declines despite some of the declines in atmospheric mercury deposition. And so a few studies have tried to answer this question of why that is. One suggestion, and based on sediment work in the Great Lakes, has been that there's been an increase in local industrial inputs, and so those lakes are adjacent to those lakes are metropolises, Toronto, Detroit, Chicago, um, are all areas where there is a lot of anthropogenic activity. And so it's been suggested that, like, with a decline in long range atmospheric transport and deposition of mercury, there's been an increase in local deposition of mercury. And then the other thing that has been noted are changes in the food web. Lake Michigan, in particular, had a quagga cladospora invasion, so an invasion of the quagga mussel, and that had a big change caused a big change in the Lake Michigan food web, which is thought to have resulted in mercury being taken up more efficiently into fish. Um, and so while there may be a decline of mercury getting into that lake, there is a more efficient bioaccumulation through the food web. So in Lake Champlain, we don't exactly have those things, though. I mean, there are smaller cities around the lake, but we don't really have like a big industrial city that's dumping or that's releasing mercury into the lake. There have been invasions. I mean, there are invasive species in Champlain, but nothing quite to the scale of what has happened in Lake Michigan and Lake Erie, where there's been a really dramatic change in the food web that would sort of explain or counteract this effect of like mercury inputs to the lake declining. And so, like, why do we see this trend in Champlain, or this apparent trend where there was quite a steep decline between the 1980s and 2011, and then it seems like something else is now controlling the concentrations of mercury in the lake. And one of the obvious things that we look to is the watershed. Champlain sits between these two mountain ranges. It has 11 long rivers that start up in the Green Mountains and the Adirondacks, this large watershed area. Whereas each of the Great Lakes individually has a watershed to lake ratio of about 2 to 1 or 3 to 1. And so we're more like 18 or 19 to 1. And so are we just like we're getting more mercury from the watershed? Is that possible? So when we looked at that, the peat core, then there is a lot of mercury from the Industrial Revolution and from previous emissions that is now sitting in that soil. So it's sitting not just in the peat bog, but in soils all around the watershed of the lake. And with increased precipitation, are we potentially seeing more runoff of mercury from those soils that's accumulated in those soils? Is it now being transferred to the lake? And is that sort of why we're seeing maybe a pattern that is more controlled by the watershed now than it was by direct atmospheric deposition? And so, and this was something that was proposed in 2016, 2017, when that increase was first observed. So there have been previous hydrologic studies of the lake where they looked at mercury inputs in the water column from the watershed, and through a mass balance model. It seems like we do get a lot of mercury from the rivers. Sorry, the rivers are a bigger source of mercury to the lake than is atmospheric deposition. But like when you go out on a small boat in the middle of Lake Champlain, those deep offshore basins are in a lot of ways decoupled from the watershed. There have also been studies on smaller lakes than Champlain that show that mercury that's deposited from precipitation is taken up very efficiently into the base of the food web, whereas what comes in from the rivers tends to be deposited in the littoral areas and maybe not make it into the deep part of the lake. And so a tool that is fairly new that we have to better understand the sources of mercury to fish is mercury isotopes. Um, I love talking about mercury isotopes. I'm going to restrain myself to a single slide here, but we did a study of looking at mercury isotope ratios in fish from Lake Champlain and then compared them to fish from the Great Lakes. And these are only the really deep water fish. So these are the lake trout, which are inhabiting the deep offshore basins of Lake Champlain and not the littoral zones. And so here I'm showing kappa 200 mercury, which is an isotopic signature of mercury. So mercury that's deposited from the upper atmosphere with precipitation has a different, or it has a much higher kappa 200 value than does mercury that's coming from the watershed that's been taken up from the air by plants and then deposited in soils and washed into rivers. And that's one of the powerful tools of mercury isotopes, is that we can tell a little bit about where that mercury came from. And in comparing, so these are all mercury isotope values of fish from the Great Lakes and from Lake Champlain. We see that Lake Champlain actually does have it has a lower kappa 200 value, meaning that it is more watershed derived mercury in those middle basins than from direct deposition. And so that does point to the lake's sensitivity, or the lake's fish's sensitivity to watershed inputs. And the mercury isotope values give us sort of a top-down view of where that mercury came from. So to couple with that, we also did a study of bioaccumulation at the very base of the food web. So I said that what happens at the base of the food web really uh, controls the mercury concentrations higher up. So if you have very efficient mercury bioaccumulation into phytoplankton and zooplankton, then that is going to carry up the food web and there will be higher mercury concentrations in fish. And so I think this was 2023, we did a study of methyl mercury and mercury concentrations in water, particulate, and zooplankton in the deep and the shallow basins of the lake. This was one of those field studies that you do where we had a great plan for it. The UVM was going to get their vessel. We were going to go out in this nice boat, and like, and then sort of everything went wrong. The vessel didn't show up in time. We had a really hard time. I think Tim Myhook from SUNY ended up bailing us out and letting us go out on his very small boats. And then this was also the year where it rained, like, I think every weekend. I rescheduled we went out six times. We went out six times total. And I rescheduled each of those trips, like, I think five times, just trying to get out on a day where the water wasn't terrible and there weren't whitecaps on the lake because we were in just a tiny little boat. So exciting to do some deep water studies from a very small vessel. When we do a water column study, we're also always concerned as to whether or not what we're getting is representative. Is it representative of a year of the lake? And I mean, I don't even know what that means anymore, but 2023 was the year we had the first of the 2,000-year storms. Lake levels were really, really high and they stayed high well into the fall. And then we had the big storm on J July 11th and 12th, and that had repercussions for turbidity and nutrients in the lake. So this is discharge from the Lamoille River, and we sampled before, right after, and then a while after. And that likely had, an effect on the concentrations of mercury that we saw in the lake. This is the first look at mercury in the water column in the hypolimnion of those deep basins in Lake Champlain. One thing that we had wondered, because methyl mercury can be formed in anaerobic environments, is in some lakes that stratify, meaning that we see temperature stratification where the bottom of the lake is really cold, but we can also see oxygen depletion in those deeper basins. And that can sometimes lead to an increase in methyl mercury concentrations in the water column. We don't see a stratification. We don't see oxygen depletion in the deep parts of the main lake basins, and that is there. There's a lot of circulation through there that I think prevents oxygen from becoming depleted. We also saw no real trends in mercury or methyl mercury with depth at any of the basins or time points, and so we didn't see higher methyl mercury in the deeper waters. Okay, tables are a little bit hard to look at, but I color coded them. So these are all of our these are all of our methyl mercury concentrations in the water across basins and time points. And everything is pale yellow, meaning that we had very little variation between basins in terms of methyl mercury concentrations in the water column. And so what that means is that because we are seeing differences higher up the food chain, that they are not caused by the concentrations at the water level. The particulate in lakes is just whatever passes through or whatever doesn't pass through a filter. The particulate in a lake is mostly the phytoplankton. So it's the phytoplankton methylmercury concentrations actually varied quite a lot temporally. So starting in June, methylmercury concentrations in the phytoplankton are high, and then over the time course of the summer, they decline. And that is likely because that's likely biodilution. So algal as algal masses become more dense, then they're decreasing the concentration of methylmercury at the phytoplankton level. So we do see across all basins, we are seeing biodilution. As phytoplankton gets more dense, there is less mercury in the phytoplankton. And then that trend increased into the larger size fraction for the shallow bays only. And so the shallow bays, Missisquoi and St. Albans, are heavily affected by cyanobacteria blooms. And so we see a lot of eutrophication in the late summer in those two areas of the lake. We were trying to get zooplankton these are samples that we collected in a net. So we were trying to get at that higher trophic level. In fact, we didn't. So because the cyanobacteria blooms were so dense, we weren't able to our zooplankton toads were mostly phytoplankton. And the concentrations in those toes are really dependent on whether that toe was mostly phytoplankton or mostly zooplankton. But overall, we had fairly low concentrations in those two shallow basins. And so that is the effect of eutrophication diluting mercury bioaccumulation. We saw a really different story though in the deep basins. And so here we took tow or zooplankton toes from the epilimnion and then from the hypolimnion. So the epilimnion being from the thermocline about 20 meters deep to the surface, and then the hypolimnion, which is where the lake trout are inhabiting, from about like in the deepest basins, it's from 100 meters to about 20 meters. And we were doing vertical tows of each of those sections. And so the trend that we saw across those deep basins was that in June, methylmercury concentrations in the plankton were low and they were consistent between the epilimnion and the hypolimnion. And then in midsummer, and this is about a week after no, 3 weeks after the storm, it appears that the concentrations in the epilimnion decline, and that's likely because we have a large amount of phytoplankton in those toes. Whereas the concentrations in the hypolimnion, so the phytoplankton are really only in the surface environment where there's sunlight penetration. In the hypolimnion, mercury concentrations increased in the South Basin and in the South Main Basin and slightly in the Main Lake Basin. And then in late summer, there are consistently higher concentrations of methylmercury in the hypolimnion and also higher concentrations than there were in June. And so across those 3 months, concentrations of methylmercury in the hypolimnion increased. The difference between the hypolimnion and the epilimnion, I think, is because we have too much phytoplankton entrainment in that surface sample. What I would suggest that this means is that there is more bioaccumulation over time in the hypolimnion samples. And so if we have a longer productive season, we would expect that bioaccumulation might keep increasing. And we actually saw the same pattern in Mallett's Bay, which is our oligotrophic bay. The only difference being that we sampled right after the storm in that one, and the water was still really turbid. But again, early in the summer, until late in the summer, we see a big increase of methylmercury concentrations in zooplankton. And so what those three things together mean is that Lake Champlain has some specific sensitivities to environmental stressors. It's particularly sensitive to mercury coming from the watershed. It's watershed inputs that are driving the mercury concentrations even in the deep lake fish. The second is that the length of the productive season, so the length of the summer, which is getting longer with climate change, is likely to increase methylmercury concentrations in zooplankton. And then the third is kind of an unknown. Like we're concerned that we're on the verge of a quagga invasion or some other invasive species, and that those can have a really big effect on mercury bioaccumulation. So those were sort of the findings of our study. Like the monitoring that the Lake Champlain Basin Program does is really important to understanding what's going on with our lake over time, and also to better understand what the stressors are that are specific to Lake Champlain itself and to the different basins of Lake Champlain. I want to reiterate what Anna said that I'm going to be only on the Tuesday because I'm teaching, but I am going to be at the Lake Champlain Research Conference and presenting on, we took PFAS measurements on some of the fish that we caught through the study. And so I'm going to be presenting on that on the Tuesday, and I would love to see people there if you would like to chat. And then I have a bunch of people to thank. The study was so much fun. So my collaborators are Celia Chin, Andrew Schroth, and Chris Stepanek. And then Saul Blochter from the UVM lab, Tim Myhook, and Luke Myers totally bailed me out with boat use. Christine Gartner from Dartmouth, Meg Modley from LCBP, Ellen Marsden from UVM all helped with fish collection, as did Ethan Rutledge from Amherst. So many thanks here. I'd love to take your questions or to talk more about mercury in fish in the lake. And so I would like to open the floor to that.